Okay, this is week 9. Our last week. Okay, this is week 9, our last plant week. So today we're going to talk about plant reproduction and plant behavior, just sort of how the plants respond to their environment. Okay, so let's start with hormones, plant hormones. So the last week we talked about how plants grow, things like that, but really there has to be some sort of chemical signal triggering them to do these things. So plant hormones are the molecules that tell the cells what to do, to grow, stop growing, to be dormant, etc. Okay? There are 5 major hormones that you need to know, and I'm just going to give you the basics of each hormone, not all the little tiny details. First major hormone you need to know is auxin. Okay? Auxin is probably the one that you will hear of the most. And primarily, the easiest way to think about it is auxin promotes growth. Okay? It's a growth hormone. It makes the fruits develop. It makes the stems grow. It makes the leaves grow, all sorts of things like that. And oftentimes what you'll see auxin associated with is something called apical dominance. Apical dominance simply means that you have a plant, let's say this is my plant here, right? It's pretty tall, and it doesn't have a lot of what we call lateral buds. It's pretty much just mostly growing tall. Doesn't have a lot of width. Okay? So auxin promotes apical dominance. You'll see auxin again later on when we talk about tropisms as well, but for now let's just move on. Okay, next hormone we have our gibberellins. Okay. Okay. Gibberellins are also associated with growth. Okay. They cause growth in the leaves. They actually inhibit the roots. And what they're also known for is cell elongation. So not only will they make the leaves grow, they'll make the stem cells get longer so that your plant gets taller faster. A lot of times what you see associated with gibberellins is a phenomenon called bolting, where you have a very, very tall plant, way taller than any other normal plant, and that's usually because it's been exposed to gibberellins. Okay, our third hormone is cytokinins. Okay. Once again, growth, and it also has to do with the development of the seeds and the fruits. Okay, so really what you can think of is these first 3 we're all looking at they're all growth hormones. Okay, the next 2 that we talk about, we're going to sort of switch gears here. Fourth hormone is a hormone called abscisic acid. Okay, this is like your plant's stress hormone. Because abscisic acid tends to act in situations where your plant needs to deal with some sort of external stressor. Let's say there's a drought or stuff like that. Okay? So what abscisic acid does is it promotes dormancy. Because when your plant doesn't have the right environment to thrive, it's going to try to be dormant so that it can sort of hibernate in a sense until the conditions become favorable again. So it promotes dormancy, and it also closes the stomata in dry seasons. That way the plant's not losing too much water. Oops. Okay. The last hormone we'll talk about is ethylene. So ethylene is a little different in that it's a gas. Okay, and what it actually does is it promotes ripening of the fruits. And it also promotes senescence. Senescence is when your leaves fall off your trees. And that's, again, both of are sort of end of the road things, right? Your plant is, on its way out in a sense, or the fruit is ripening, everything's sort of ending. Your plant's not growing anymore. Ethylene actually has a sort of interaction with auxin in that once you have a certain amount of auxin, then ethylene starts being promoted to sort of start that end part of your plant's life. Okay. Senescence, that's a term you want to know. Again, that's when the leaves are falling off the trees. Usually that's either for dormancy purposes or unfavorable conditions. Okay, so now we're going to look at tropisms. Tropisms are basically a plant's response to something in the environment. Okay, the first tropism we're going to look at is a plant's response to light. That's called phototropism. Okay. And basically, what they found is when a plant was grown such that a light was placed on one side, so one side of the plant got more light than the other, the plant would grow towards the light. Okay. So let's say this right here is my lamp. That's the light. And I have this shoot right here. Okay. Over time, what they found was the plant would start to curve towards the light. Okay. And after they did some experiments, what they realized was that auxin is the one that is actually causing this to happen. Because if I was to look at this shoot in more detail, let me get closer up, right? If this is my light over here, well, auxin actually responds to light by transporting itself to the shadier side. Okay. So by doing that, since auxin is a growth hormone in shoots, what happens then is this side of the plant that has more auxin is going to grow more than this side of the plant, which doesn't have auxin, okay, or not as much anyway. So what you end up with is differential growth. This side is growing faster than this side, so it ends up curving. This side is longer, ends up curving towards the light. Okay. Gravitropism, or what you might also see written as geotropism. That's a plant's response to gravity. Okay. And basically what they found is if a plant, let's say, we're going to have to do it differently for roots and shoots here, but for a stem, okay, if they were to lay the stem on its side like this, okay, the stem would still start growing upwards like that. The root would start growing down into the ground like that. Okay. Either way, though, they determined that it had to do with auxin. Okay. So auxin, here's what happened. They think there's another molecule involved. They're called amyloplasts. So amyloplasts are these starch-containing vesicle type things in plants. What happens is amyloplasts would be drawn towards the ground, so they migrate towards the ground in response to gravity. Okay? And when they did that, they're kind of heavy. What they would do is they would push on the endoplasmic reticulum. Okay, so basically you've got now, let's say in our two different samples here, right? Your amyloplasts are all down here. Okay? They're being weighted down by gravity. They actually trigger auxin to be released at that location where they are, because it's thought that they press on the endoplasmic reticulum, makes the endoplasmic reticulum release some ions, and that triggers auxin to be released as well. So it triggers auxin release, which means now I have a lot of auxin being produced down here and down here. Okay? So once again, because of that auxin, now you're going to have differential growth. In a shoot, okay, that's going to promote growth in the shoots. Okay, so this side right here, this side of my shoot right here is growing more than this side of my shoot, so that's why it starts curving up, because this side's growing faster than this. In my root, okay, it actually inhibits growth. So in this case, this side right here is growing less in my root than this side right here. So that's why it starts growing down into the ground. Okay. One last quick tropism to mention, but I'm not going to go into any detail on it, is thigmotropism. That's a response to touch. A good example where you might have seen that would be like, tendrils growing around a trellis or something, or Venus flytraps, things like that. Okay. Okay, one more response to the environment that we need to talk about is photoperiodism. So photoperiodism is basically, it's a response in terms of flowering. It's flowering plants. Some of them will flower when they're short days, some will flower when they're long days, but really what it ends up meaning is that the flowering is dependent on the length of the day or night. And I'll explain what I mean by that in a second. Okay, first let's talk about the three types of plants you're going to have. First of all, you're going to have short day plants. Okay, they flower when days are short. You have long day plants. They're going to flower when days are long and nights are short. And third, you have what we call day neutral plants. They're going to flower regardless of when they don't care how long the day or night is. Okay? And simply put, what they've figured out here is that these plants are basically regulated by a molecule called phytochrome. So really, phytochrome is what determines sort of when a plant will flower. Okay. There is also possibly a hormone called phloragen that actually controls the flowering, but the phytochrome is supposedly the trigger that's sort of turning on the flowering or turning off the flowering, etc. Okay. Phytochrome has two different forms, the PFR and the PR. And basically, depending on what kind you have and how the plant responds to it, that's how it's going to decide if it's ready to flower. Okay. What they also realized, though, in their experiments in looking at photoperiodism is it's actually not the length of the day. Okay. It's actually the length of the night. So it's the amount of night or darkness is the key to flowering. Okay, but they also call it something called critical day length. So they used to think it was daytime, so they named them all with the day names, but really when you get down to it, it's the night. Okay, so that's pretty much it for plant behaviors in terms of how they're responding to their environment, etc. Now we're going to change gears and move into plant reproduction. Okay, so before we start talking about plant reproduction, it's important to sort of realize that plant reproduction to a large extent also defines the phylogeny of plants. Who is more advanced than another plant? Okay, so if you remember here, if we have a common ancestor, probably some sort of green algae, then the first branching that we'll have is the non-vascular vascular. So these would be your non-vascular plants. Okay, and within the vascular plants, then you have a division. First you have seed versus non-seed plants. So right here, the first seed, the first vascular plant would form. These are going to be your ferns. They are non-seed plants. Okay. Further up the line, you're going to have another dichotomy here where you have your gymnosperms and your angiosperms. Okay? So in terms of reproduction and what makes them more advanced, well, first of all, these are non-seed. These two are your seed plants. Okay? So these are going to be the most advanced right here, because having a seed really allows for a greater chance that the embryo is going to survive to grow and propagate and pass on its genes. Okay, ferns, no seeds, they use spores. Mosses also, well, they don't have vascular tissue, they're even less advanced. Okay, so what we're going to focus on mostly in plant reproduction is angiosperm reproduction, but dealing mostly with these seeds and why they are such a benefit, why they're such an evolutionary advantage. Okay, now, in any plant, seed or non-seed, you're going to have something called alternation of generations. Okay. Basically what that means is for any plant, if you're looking at like the plant's life history, there are two stages that this plant's, that a plant's life history will go through. Okay? So plants can exist in two stages, and they're independent stages. A plant can be a gametophyte, which is a haploid stage, or a plant can be a sporophyte, which is a diploid stage. Most of the plants you see out there, you're probably looking at the sporophyte generation. Okay, so this would be N, this would be 2N. Okay? Gametophytes ultimately are forming from spores. Okay, so we're going to look mostly today at angiosperm reproduction. Okay, let's start with structures. The angiosperm, in order for it to have reproduction occur, its reproductive organs are really all housed in the flower. So we're going to go through first the parts of a flower. Okay. So first of all, you got your petals. Petals are thought to be possibly attractants for, bees, other organisms that might help pollinate, help that fertilization process take place. Okay, but in the center of your flower, first of all, you're going to have this thing right here. Okay, down at the bottom of this bulb, this is the ovary, and the ovary contains the ovules. The ovules are going to be the units that eventually develop into the female gametophyte. Okay, now the pathway to the ovary is called the style, and the top of the female reproductive organ here is called the stigma. Okay. Now surrounding that, you're going to have the male parts. Okay, these are called the anthers, or the filaments. Okay, so the anthers are at the top here. These stalks that lead up to them, these are called the filaments. This thing at the top here, called the anther. And the entire male organ is called the stamen. Okay? So basically, when you have a flower, your male gametophyte is going to be produced from the cells at on the stamen. Your female gametophyte is going to be produced from cells inside the ovary. Okay, so in terms of the gametophytes here, the male gametophyte in an angiosperm is the pollen grain. So it's not the anther itself. The anther itself is diploid. Okay, but some of the cells on the anther will go through meiosis and become these male gametophytes. There's a little things called microspores. And so the pollen is really the male gametophyte. The female gametophyte is going to be in the ovary, in the ovule, and that's going to be the embryo sac. Okay, so let's talk about how is it then that an angiosperm is able to reproduce. So let's talk now about the whole actual fertilization process. Okay, that has to really start with pollination. Pollination, simply put, is when the pollen grain gets transferred from the anther to the stigma. Transfer. Because otherwise it won't even have access to the female gametophyte. So it's being transferred from where it was produced, the anther, to the stigma, which is the opening, if you will, to the female gametophyte. Okay? So what happens is this. The pollen grain is already haploid. Right? It's N. When it gets transferred onto the stigma, for whatever reason, it could be a bee that takes it there, it could be the wind that blows it there, lots of different ways pollination happens. It could just fall on there. A plant can actually self-pollinate, you know, the pollen from its anther can drop onto its stigma. But basically, once it does that, then it's going to be triggered to develop further. Okay. First thing that happens is that pollen grain goes through mitosis. And it produces now 2 sperm nuclei. Okay? Now each of those sperm nuclei is still haploid. Okay, still N. But what happens then is it also starts to dig its way down to the female gametophyte. So now it starts to grow a pollen tube. Okay, so it looks something like this. Originally our grain was just like this, right? The pollen grain now starts growing a little sort of projections called a pollen tube. The sperm nuclei are going to be in there. Okay? And what the pollen tube is really doing, though, is it's sitting on the stigma right here, right? This is our pollen grain where it landed, so it starts growing the pollen tube down into the ovary, this is my ovary, to try to get to the embryo sac. Okay? So it's trying to dig its way in. So it grows a pollen tube to get to the female gametophyte. Okay? When it gets there, it's going to enter the female gametophyte through a little opening called the micropyle. Okay? And after it does that, that's when the actual fertilization takes place. Now, inside your ovary, okay, let's look and see what happens in there. So this is your ovule, your embryo sac. There's actually a whole bunch of different nuclei for some reason. Okay, but within your embryo sac, you're going to have two polar nuclei, and you're going to have your egg cell, as well as an assorted other nuclei that we're not worried about right now. Okay, so that's right here. This embryo sac is also formed through meiosis within the ovule. Okay, so when the pollen grain gets into the embryo sac, you're going to have what we call double fertilization. Okay, number one sperm nuclei is actually going to fuse with the egg cell to make your diploid zygote. So one sperm nuclei plus egg cell gives you your diploid zygote. That's really going to become the plant. That's like the plant embryo. Okay. The second thing that's going to happen, though, this is why they call it double fertilization, is the other sperm nuclei is actually going to fuse with the two polar nuclei. And what that produces is something called endosperm. Okay? This is really important because the endosperm is actually going to be nutrition for the embryo as it grows. It's a starchy kind of substance. So your embryo needs food as it's getting bigger in your seed. That's the key here. Okay? So with this double fertilization having taken place, then you have your seed, and the seed begins to develop. So the seed coat forms. Your fruit begins to develop. Now is the time where you have an embryo, you want to nourish it, you want to protect it. Okay, so to do that, the seed grows a harder layer on the outside, and the fruit, which is basically the ovary, protects it as well. Okay, so angiosperms are really significant, more advanced because they have these adaptations. The fruit that's protecting your seed. Okay. Let's look at the seed really quickly in terms of its components. So, first of all, one important thing I want to mention here is a lot of people get gametophytes and seeds confused. So, a seed is not a gametophyte. Okay, what a seed is basically the zygote, the embryo. So it is what? Diploid or haploid? It's going to be diploid. Okay? It's basically the stage before the embryo has fully come out of that protective layer, and it's beginning to develop into what will be a full-fledged plant. Okay, what will be a full-fledged sporophyte. So, your seed, there are three main parts you need to know about the seed. Number one, you have the seed coat. Literally what that does is it protects the seed from the outside environment. Okay, from drought, from fire, from whatever it might be. It's a protective layer. Okay. Within your seed, we also have the embryo itself, and part of the embryo is going to be something called a cotyledons. Okay. A seed will either have one cotyledon or two. We'll talk about that a little later. Third part, the endosperm. Remember, that was formed from the polar nuclei and one sperm nuclei fusing, so that we get this triploid, right, 3N, really, substance, and that's going to be for nutrition. Okay, so couple other terms related to seeds. Germination. That is when the seed begins to develop. And ultimately, the cotyledons grow, they break through the seed coat, and that's when you have your little baby plant beginning to form. Okay? The coleoptile. That is basically a leaf sheath, like a little protective leaf. And what it does is it protects the cotyledons as they, or the beginning, whatever the first leaf is, to sort of break through that seed coat and break through the soil. It's going to protect the first leaves as they go through the soil so they don't get all damaged as they're pushing through. Okay. So, one thing we're going to talk about related to plants and introduction here is we talked about the cotyledons. And if you remember, there are two types of flowering plants. There are monocots and there are dicots. And what that directly relates to is their cotyledons. Okay? So let's do a quick comparison of monocots and dicots. Okay, first of all, the name itself tells you how many cotyledons each of these is going to have. A monocot, mono means one. Cot is just short for cotyledon, so it's going to have one cotyledon, one seed leaf. Okay, dicots, di means two, so it has two cotyledons. And there are other ways that you can recognize monocots and dicots. Number one, you could look at the veins in the leaves. Monocots have parallel veins in their leaves. Okay, dicots have like a branched or net-like pattern of leaves. Okay, if you were to look at the number of flower parts, for example, the petals, you would find monocots have flower parts in multiples of 3, 3, 6, 9, etc. For dicots, you're going to have flower parts in 4s or 5s. Okay. Last one is if you were to actually cut open the stem, looking at a cross section at the vascular bundles, the xylem and the phloem. With monocots, you would see scattered vascular bundles. They're sort of all over the place, not in sort of particular patterns. In dicots, you're going to see vascular bundles in a ring form. Lastly, quickly, let's just talk about gymnosperms because they are also seed plants. I just want to mention how they are different from angiosperms. Gymnosperms, they are seed plants. They have what we call kid seeds because their seeds are not protected by any sort of fruit. No fruit. Okay, but they do then are cone. They are conifers. Okay, So they're going to end up carrying their gametophytes on cones. You have male and female cones. And then ultimately, they will also go through a reproduction process, not unlike that of angiosperms, but in this case, you won't have, the, pollen grain digging its way through and things like that. So that's about it. That's it for plants. Bye-bye.